Welcome to the Universal Sequencing Technologies first Q&A, a free Q&A webinar that we'll be doing weekly. And we'll go through a quick introduction of the company and some technology that we'll be talking to on a regular basis. And then we'll go into the Q&A. And the Q&A will be managed by David, where the question will be then forwarded to Andrew and or Tom or myself to respond. Please, if you have any questions, even before we start, you can just send them on the chat, and we'll get started. And basically, the introduction will be 5 to 10 minutes, and the rest of it will be Q&A. Okay, let's go ahead to the next. By the way, before we start, this is our new building we recently moved to in Carlsbad. It's about 20,000 square feet facility, and that's where all our R&D, the laboratory, the development, engineering, and administration staff, including sales. Andrew, next. Quick introduction. Universal Sequencing Technology provides innovative low-input, super long-read DNA sequencing solutions. We call this our TEL-seq technology. Simply turns a second-generation short-read DNA sequencer into a third-generation long-read sequencer. The affordable TEL-seq library kit is used in microbial genome assembly, metagenomics, de novo assembly, whole genome phasing, phase whole exome, and target sequencing and structural variant identification. We provide the kit and the analysis pipeline, and all you need are the samples and some common lab equipment. Basically, as a summary, before we start here, the library prep kit generates link read as follows: a simple one-tube, fast and easy to use link read library kit, converts short read sequencers into highly accurate long read sequencers, and it wears very low input material, 0.1 to 5 nanogram DNA. Applications, as we discussed before, are outlined here below. The important thing is that it's a combination of what we do is we provide the library kit to work with Illumina sequencers. And that's a partnership that we have with them as a co-development, co-marketing relationship. Next. All right, Andrew, give a quick background and then we'll go to the Q&A.
Okay. Hi there. For those of you that don't know me, I'm Andrew Anfora. I am the Senior Field Application Scientist Manager for Universal Sequencing. So if you have questions during or after this Q&A, I would likely be your main point of contact. And I want to just give you a very brief background for those of you unfamiliar with TEL-seq, just to kind of ground some basic descriptions. So want to walk really quickly through this cartoon. What we see is a diploid genome where there are one chromosome has its haplotypes colored in blue, the other chromosome has its haplotypes colored in red. As you're probably aware, with short read sequencing from Illumina, you get highly accurate reads, but they're short, so it makes it very difficult to phase a genome or prepare a de novo assembly. Long read sequencers such as PacBio or Oxford Nanopore give moderately long read lengths, either slightly shorter lengths at high accuracy or slightly longer lengths at lower accuracy. And these allow for some basic phasing and more robust de novo assembly than plain short reads. What TEL-seq is a linked read product where we are going to be tracking the intact molecules before fragmentation and attaching a unique identifier to the molecule, such that when we produce the library fragments, we're able to trace back fragments to the same molecule of origin. And this lets us preserve size information and capture information for molecules greater than 100 kb in length to stitch them together and allow for both phasing and de novo assembly of a genome using a short read sequencer, so you get that good Illumina accuracy with the ability to span larger content. It is a very fast workflow. From isolated DNA to sequenceable library takes 3 hours. If you've ever done any sort of NGS library prep, you would be able to probably master this the first try. It's a very straightforward prep. So with the 3-hour library prep turnaround time, you could go from isolation of DNA to collection of data within one workday. We provide software to either assemble the genomes or phase the genomes. I don't want to go into this diagram in too much detail. Just the green on the diagram is the freely available and provided by universal sequencing software options. These software are also are on base space. Since a number of people that have used link reads in the past really like 10x genomics software, we do also provide a conversion tool to convert our data format to a format compatible with 10x genomics software tools, and that is what is depicted in blue on this chart. Again, our software is freely available on the Universal Sequencing website, or if you're a base space user, you can run our software through base space. Just to kind of give a bird's eye view summary then, it is a 3-hour library prep protocol, so nice, quick, and easy library prep with super low DNA input. So microbial genomes and targeted panels are looking at sub-nanogram input, and then a larger genome, like a human genome, only requires 5 nanograms of DNA for the library prep. And we're able to preserve information for molecules greater than 100 kb in length. So really long, even by linked read methodology standards. And our software is freely available, as I mentioned. And as Aaron alluded to, we are already global, have customers the world over.
Thank you, Andrew. And Tom and Andrew, could you present your position in the company one by one, and then we'll proceed with the questions. Andrew?
Sure. So I am the Senior Field Application Scientist Manager. So I am the main touch point for technical questions and for users that have already used our materials. If you have a technical question, I am in charge of manning our tech support email, or can be contacted directly, and really am the first point of contact for technical issues.
Thank you. And Tom? You're muted.
So sorry, I'm muted. Yeah, this is Tom Chen. Yeah, I'm the CSO of the company and also developer of TailSeq technology. I really am so glad to have this platform and opportunity to directly communicate with our current TailSeq users and the future TailSeq users. Yeah, all I want to say is, yeah, we are here to support you. Yeah, if you have any question, please feel free to ask us. Thank you.
Thank you, Tom. And David?
Good morning, everyone, or good evening, depending on where you are. I'm David Nordell, the Inside Sales Manager for Universal Sequencing. I'm your primary point of contact for quotes and purchase orders, and I'm sure you all already know me. So if you have any questions regarding sales or purchase orders, feel free to contact me after the webinar.
Thank you, David. And myself, I'm the Chief Commercial Officer and General Manager at Universal Sequencing Technologies. And it's a pleasure to have you all together here in the morning or evening. And we'll open the platform for questions. So let's hear if we have anyone with some questions. We'll wait a few minutes, and if we don't get any questions from others, do you have anything, David?
Yeah, so I have some stuff. So for one of the questions that we seem to be frequently getting recently is for legacy 10x genomics customers that were using their linked read technology. How can TelSeq function as a replacement for that technology that's no longer available?
Sure. So I'll take the first pass, and Tom, feel free to fill in if there's other things you'd like to add. So I'll actually go back to our slide with the software. So we generate linked reads that are similar to 10x genomics in that the parent molecule is tracked with a unique molecular identifier. The major difference between the library structure for us and 10x is 10x uses a 16 base pair barcode. We use an 18 base pair barcode. So we provide a software tool that has to be run to convert our data format into a 10x compatible, and then our data can be run directly into Long Ranger and or Supernova once that conversion tool has been applied. So our data are highly functionally similar to the 10x linked read data.
Yeah, I just add a couple of points. As Andrew mentioned, yes, it's for TLC, the functionality is very similar to 10x in terms of result. In addition, TLC has the advantage, which is our reaction is much easy to scaling up and down, but based on the target, it's not like 10x just always fitting for a large genome. So by doing that, TLC can easily to do the small genome and also to the targeted application with reduced cost. Yeah.
Great.
Thank you, Tom.
David just mentioned that the microphones are enabled, so you can ask the questions directly. I will ask Andrew and Tom to make the following comment. With the combination of UST, TelSeq, and Illumina sequencers, how well the combination competes against key competitors like PacBio and the Oxford Nanopore and so forth. Any comment on that? Why is it better, the combination of TelSeq and Illumina against the other competitors?
Tom, would you want to start, or do you want me to take this one first again?
Yeah, you go ahead.
Okay, so really what we're doing is you are getting the base pair calling accuracy and cost per base pair of data acquired that you would with an Illumina system, which compares favorably to true long read technologies, but we are still able to span with a high degree of certainty those super long DNA fragments. So we're still giving you positional information for molecules, tens of kb, hundreds of kb in length, but within the Illumina workflow that is more familiar and has really excellent base calling capability.
Thank you, Andrew. Okay, we have next question from Frederick. Are your applications linked to NovaSeq, or can we switch to Nanopore Minion?
So for now, our commercial workflow is optimized around Illumina instruments that can take spike-in sequencing primers. So our assay can work with any Illumina system except for iSeq out of the box. And Tom can maybe comment. I think we're still determining what, if any, other sequencers we will be officially supporting. Currently today, our off-the-shelf product supports Illumina sequencers except for iSeq.
Tom, you want to add to that?
Yeah, it's our initial development is mainly for the short-read sequencer, basically for Illumina sequencer. And in Asian countries, there's some customer also use on the MGID instruments. That should be okay. For the other technology, is at this moment it's not available, but internally we're looking through some potentially in the future development. Yeah.
Okay, great, thank you. All right, next question we have is from Khalil. Do you have experience using TelSeq in polyploid SPP?
Yeah, probably let me answer this one. Is it's again currently our software design is for deploy the genome, and which is working reasonably well. For polyploid samples, we do see there's some publication use 10x link read. They can with special analysis solution, they can do some pretty reasonable analysis. At this moment, yeah, we don't have a good internal software solution on the polyploid sample yet. However, we work on the field, work on the people, talented bioinformatics people to look into this. So theoretically, yeah, it's workable. Practically, yes, you indeed need more software development to fully support this application.
Thanks, Tom. All right, from Chris, we have, do we need to use different amounts of TEL beads for different genomes, i.e. microbial versus mammalian?
So yes, in our user guide, we outline 4 different reaction conditions based on expected genome size. So we have default guidance based on genome size, depending on the genome size. If it's especially like intermediate organisms, there might need to be some optimization, but we give you really the 4 good starting points that seem to work well for different genome sizes. So we try to build a lot of flexibility in the kit. So the number of reactions you get out of a kit is going to be inversely proportional to your genome size.
Yeah, and also recently we have more and more users interested to look at a smaller target, such as the 10 kb amplicons, or sometimes even smaller. So we developed a protocol to just dealing with this kind of application. Yeah, we should roll out this app node and the associated protocol in the next couple of weeks.
And that kind of leads into the next question. How many genome samples can you process with the standard TEL-seq kit of various genome sizes? So the range there, on the small genome size to large genome size.
Sure. So we have 2 kit formats. We have a standard format, and that kit configuration is enough for 12 microbial genomes, or 4 human-sized genomes, or an intermediate number of intermediate sized genomes. And then we have a higher throughput kit for larger projects that can handle 24 human genomes or 72 microbial genomes. And the other multiplexing parameter is we have library-specific multiplexing primers, of which we sell 3 sets of 8. So you can multiplex up to 24 samples on a flow cell.
Thank you. Okay, and a follow-up question from Chris. What are the recommended Illumina machines and kits for sequencing for microbial versus mammalian genes?
So the short answer would be whatever instrumentation you like best. We typically recommend collecting read lengths of 2 by 146 paired end. Apart from that, it's really whatever sequencer you think makes the most sense for your project. And that's something we could discuss in more detail offline if there is a specific project concern.
And those are all the submitted questions we have so far.
Andrew and Tom, let's say describe a quick example from the industry of the experiment that Dr. Hernandez performed on insect, and give a quick outline how this TEL-seq would use with Illumina sequencers to accomplish their project.
Tom, would you like to outline that?
Sure. I think probably you guys are already aware, one of the key advantage for TailSeq application is we use a very little amount of input. So it's again depend the genome size. For the smaller genome, we can use as low as even 0.1 nanogram, but quite often our standard 0.5 nanogram for smaller genome, which including some microbial or any genome less than 200 megabases. For large genome, we use 5 nanogram. So this go back is for some insect applications in the Earth genomics. Some of the sample is immediately issue is really hard to just get enough genomic DNA, especially it's sometimes so important to only get a DNA extraction from a single insect. So that is truly is a fully take advantage. TailSeq can offer for those samples with rare, very small amount of DNA input. Is our collaborator is the UIUC, Dr. Hernandez lab. They actually successfully be able to get enough DNA extracted and make a TailSeq library. That sequence get pretty good de novo assembly. So that's a way it's been demonstrated in the field for TailSeq for the low input. It's potentially currently the best platform we can use. In addition, and these days there's a lot of options in the field. Also, there's a direct long read platform like PacBio and ONT, and also we offer link read on the Illumina. So the people also do the combination approach, and if they are affordable, they have the PacBio platform, and they have the funding. They can combine the PacBio the long read to build up the initial platform. That with a TailSeq. Again, another advantage for TailSeq is we can offer much longer range the sequencing information with good high molecular weight DNA, and we can get the link read molecule length quite often over 100 kb. So that's it's not the long read technology can easily provide it. That's why there's a user already demonstrated they can use PacBio the short, relatively short 10 to 15 kb. Early days we never saw this would be short. Yeah, they can use a 10 to 15 kb long read with our over 20 to 100 kb link read molecule information to further scaffold significant input final assembly result.
Great, thank you, Tom. Anything you want to add, Andrew?
I think just to reiterate, so our low input means that it's practical to do a single organism, quote unquote, personal genome, even if you only have a very small insect. If you can get any DNA out of a sample, you can use our product. And for projects where biomass is limited, then you can still get a usable de novo assembly using only our technology. And as Tom mentioned, since we have those 100 kb plus read lengths, you can still further scaffold a pretty good PacBio or what have you assembly. We have both those very long fragments, and also since we are a different technology, there's just going to inherently be different breakpoints in the assemblies. So it really can make a pretty good assembly better or is great on its own.
Thank you, Andrew. And David, do we have any more questions?
Let's see, we have a question. Any success in de novo application in ocean life sciences? If yes, can you elaborate on the details?
So while Tom is thinking if we have any specific examples, I would just say if we're talking about just higher marine organisms, if it has DNA, we can sequence it. We are still working on some metagenomics tools. So I would say if it's a low complexity microbial community that's the marine area of interest, it would also be feasible. We're still building out our guidance on both complexity and contamination handling for complex microbial communities. So kind of depends what aspect of marine community organism the interest lied.
Thank you.
Anything else, David? From the chat?
That's it for now.
That's it for now. Well, if there are no more questions, I would like to thank you for joining us today. Again, we'll be doing this weekly, every Thursday, the same time. And if you prepare with some questions, we'll be glad to answer you. And again, thank you and wishing you a great day and evening.
What? We have someone typing. Maybe they're putting a question in real quick. One second.
Okay, great.
All right.
And David, you enabled their microphone already, right?
I did, yes.
Okay, so they can, anyone wants to verbally, instead of typing in the chat, you can just ask and we'll hear you.
I guess we'll go ahead and start wrapping up then. If you do have any further questions, you can reach us all by email. I will go ahead and put Andrew's email in the chat for you to be able to contact him directly. And I will put mine in the chat as well for any questions or purchase orders. Once again, thank you all for coming, and I'll hand it back to Aaron.
Thank you very much again, and we'll see you next week.
Thank you.
Hey, bye everyone.
Thank you.